Cool, I still have a pointer. Awesome, okay. Thank you guys for bearing with me through some technical difficulties. Hopefully this won't happen later. So I'm going to be giving the next 2 lectures about X-ray crystallography, since this is kind of what is my bread and butter, kind of basic. Every day I'm thinking about crystallography in the lab. So feel free to stop and ask questions as I go through. If I don't notice you, just keep waving at me and I'll stop and we can talk about it. But a lot of this information is a little, actually I still want my notes on my computer. Sorry about that. Yeah, just let me know if you have any questions as you go along. A lot of this is not intuitive, so feel free to stop and ask questions as we go into a little bit deeper on X-ray crystallography specifically. So specifically in X-ray crystallography, there's kind of like a workflow. So it always starts with our extremely pure and homogeneous sample of either protein or a different macromolecule, like as a polysaccharide or RNA or DNA. And then you are able to crystallize it. Once you capture your crystals, you irradiate it with an X-ray beam at either a home source or at a synchrotron, like we've talked about earlier. And then we take the detector's, the diffraction pattern that we've been able to capture on our detector, and then compute the electron density from the resulting diffraction pattern. That electron density is then used as a model that we can build our molecular scaffold into to actually create the really beautiful structures we see on the PDB. So you have your pure protein, you get it into crystals, diffract it, you get these maps, and then you fit it, and then you go back and further refine your diffraction until you get a beautiful model at a specific resolution.
So how exactly do you use the diffraction pattern? Like how do you...
We will talk about that.
Okay.
So one thing that I think is really important to know about this is that there's a lot of work that goes into the process that is before crystallization. So initially, you may have a protein target that you're really interested in, but you're not sure exactly which one to use. So most proteins are produced in E. coli using a cloning-based system. Excuse me. Sorry about that. So this, using a cloning-based system, so you have to choose a protein that is able to be effectively expressed in E. coli. So this involves a lot of different bioinformatic analysis, such as codon optimization, because certain codons are not used in bacteria that are commonly used in eukaryotes and especially mammals. So oftentimes people who are interested in proteins that are present in humans will actually choose a bacterial homolog or a very sequentially or structurally similar protein that's actually from a different organism, so it can be easily expressed and then overexpressed in the system that they're using. Then you have to actually create these recombinant vectors, which you can see part of a plasmid right here, through the cloning process, and then make sure you've sequenced that effectively, you have exactly the DNA sequence you're looking for. Then you have to go through the sometimes long and arduous expression purification process. So you have to be able to overexpress your protein at a level where it is still soluble, so you're actually able to pull it out of the solution for later purification steps. And then you have to make it super pure. So you have to go through a variety of different chromatographic methods like size exclusion chromatography or anion exchange to remove all the other kind of contaminating proteins that can often be co-purified. And then you move into crystallization, which seems like the easy part. You just put your protein into a crystallization solution and voila, you get crystals. But it's actually not that simple, and a lot of projects die here. So we actually have had a lot of problems with proteins in general being crystallizable. So sometimes you go through these steps and you try and crystallize and you try and crystallize and you try and crystallize, and you have to go back and go, okay, well, X construct was not crystallizable. How can I try and make it more amenable to this process, or are there other homologs I can look at instead? Then once you do have crystals, you look for diffraction data through the screening process. So you send them to a synchrotron and see if you're able to get high resolution diffraction data. And if you do, you collect that data and then move into the bioinformatics again, and you start using a variety of different programs to actually determine the structure, validate it. And then you can potentially use these structures for drug discovery or structure-based drug design. Oh, come on. There we go. Yes, question.
Have people ever used human cell lines or another kind of cell line for purification?
You can.
Express the protein.
So it's more likely that people will use insect cells because those are a eukaryotic cell line, because they don't have the requirements of mammalian cells. So mammalian cells not only have oxygen requirements, they also require specific nutrients, and you can't grow as much of them. And they don't really like to overexpress things because that makes them, they think they have cancer, and so they die. So it's actually really, really difficult, and it requires way more samples, way more time, and you end up with much less of that very pure protein. But it has been done. It's just typically they would rather use yeast or insect cells for those kind of more fragile proteins, especially if they need chaperones to fold correctly. You typically move them into an insect cell line or a yeast cell line, because those are more amenable to the overexpression process. Oh, I'm still one slide ahead. There we go. Okay, so we're talking about diffraction for the most part, but it would be much easier if we just had X-ray microscopes, but this is not possible. So we know that X-rays have a wavelength of 0.1 to 100 angstroms, and visible light is between 400 and 700 nanometers. So the resolution is limited by wavelength. So we can't see anything smaller than 400 nanometers with white light, but we could technically see stuff with X-rays at the atomic level. But to get direct images from X-rays, we need to be able to focus it. But we can't focus X-rays with lenses. And that's one of the key stopping points that prevents us from being able to actually create an X-ray microscope. So we bypass this with X-ray crystallography, and we actually have a defined X-ray beam, and we're able to use the regular arrangements of molecules into a lattice to give us this diffraction pattern, and we can kind of work backwards from the diffraction pattern to get that initial image that is within our crystal structure. So if we think about lenses in general, so let's pretend that this is a microscope here. We have our sample of interest, we have light shining onto it, then we have a refractive index of air, which is different than the refractive index of the glass that makes up the lens for our light microscope. And that allows us to actually change how big we view an item through the lens. So this is how light microscopes work. However, because lenses require this difference in refractive index, we're not actually able to do this with X-rays, because the refractive index for X-rays is almost exactly 1 for all materials. So there's no difference. It is equalizing the it disallows this difference between 1 and 2. So if we hit a lens with an X-ray, it's just going to show us our lens. So it's not going to actually give us the information we need. So
there's a question.
Yes.
Hi.
Yes, sorry.
I just have a question that, is there a reason why the reflective index is the same?
It's because it's at the atomic level. So if you think about this, glass is made up of atoms. And if we're looking at trying to determine the atomic structure of something else, we can't put that through another lattice of atoms and still think that we're going to be able to see it. So it's because of the wavelength being so short. So one of the reasons I think this is really interesting is with cryo-electron microscopy, because we're actually using electrons directly to detect the structure of your sample, we're able to actually focus those with magnets in a way that is very similar to a lens. So you're able to kind of create a makeshift lens by focusing the electrons, but we can't do this because X-rays are just a wavelength of light that is actually produced by the movement of those electrons within the synchrotron or within your home source. So there's nothing for you to focus besides light that you can't focus with a lens because it would actually diffract off of the lens because it's made up of the same things that you want to look at. So if we move forward and we think again about that light microscope, we actually know that there's actually two Fourier transforms in a light microscope. There's one that's the scattering of the light off of your initial sample, and then a second Fourier transform that's applied by the lens that basically deconvolutes these scattered waves into a larger image of our sample. So with X-ray crystallography, where we have no lenses, we have to actually apply Fourier transforms to get these atomic images from our diffraction pattern. So we actually are able to use this mathematical relationship to take this scattering data, which is what we would get from if we imagine these rays as incoming X-rays, and then we have our diffraction pattern that's created. Then we have to determine the correct Fourier transform to actually get to our image. So if we consider the wavelength that are so the wavelengths and the constructive and destructive interference within our samples, we're able to use the way our X-ray is actually diffracted off of our crystallized molecules to insert a Fourier transform into this interaction. You can actually do this with a couple of other things. So there are particles that can be accelerated to speeds sufficient to bring their wavelengths to the angstrom range, which is neutrons and electrons. Neutron diffraction can work like X-ray diffraction, but there are still limitations to that, and there are no lenses for X-rays. So we use this mathematical equivalent of a lens to get us back to that image that we're looking for. So let's go and talk a little bit more about the actual process, specifically about crystallization and the crystal formation process. So there are a few questions that I think are really important to think about this in this process. Is why do we actually need crystals? Why does diffraction arise? And then why is electron density not an exact GPS for the structure? And then eventually we need to talk about how we calculate electron density from the resulting data we get from our crystallographic experience. So I'm actually going to swip forward through two very interesting quotes that are here by a famous crystallographer, Randy Reed. But the gist of this is that it's actually a map of the distribution of the electrons of the molecules, not the exact molecules. So we actually have to build into these electron density maps what we want our exact atomic positions to do. So we don't actually get a direct image from the crystallography we do. We end up with this map of the way it takes up space, and from that we can interpret where things are most likely to be, which is why refinement is so important for X-ray crystallography. Here's another very interesting quote that I encourage you to go back and look at later. So why do we need crystals? Does anyone have any idea of why we might need crystals to actually do these kind of X-ray experiments? Actually, I'm going to ask someone who hasn't.
The structure becomes more uniform if you crystallize the sample.
Okay, that's a really great answer.
More organized?
More organized structure?
Well, if it was not crystallized, it would be all sorts of arrangements, and then you would just have tons of different data that you would be able to go around.
that's the key. I think everyone was saying the same thing. It's by organizing the molecules, we're able to actually see them through the 3D lattice. So the whole idea is that these molecules are able to arrange themselves in a three-dimensional fashion where there creates repeating units, and this amplifies the signal. So the more uniform your crystal is, the more consistent the orientation of all of your molecules are, the stronger your signal will be, which means when we are looking at the diffraction patterns mathematically, we're actually able to get to our real electron density much easier. So this is one of the reasons you couldn't just take, say, your protein sample in a Eppendorf tube and get a angstrom level 3D structure of the atomic bonds, because we need this repetition of symbols to actually, repetition of signal and this amplification of this constructive interference to actually determine the structure. So single molecules scatter very, very weakly, and most X-rays will pass through a molecule without being diffracted. It's only through these highly ordered structures that we're actually able to get diffraction that we can measure. Come on. Yeah. So we don't look at single molecules with X-rays. We look at these three-dimensional repeating with 10 to the 14th molecules within these crystals that are actually able to reinforce the signal that we see with each one. So how do we attain crystals? So this is something that I think about a lot because, again, I told you many projects die here where we're not able to actually create crystals. So here are the two main methods that are used for crystallization. So we have the hanging drop method seen here. So you have your highly concentrated protein or RNA of interest, also nucleic acids or polysaccharides. I'm just going to say RNA because that's what I typically work with. So you have your really, really pure RNA, and you pipette that onto a cover slip, like you would use for a microscope, and then you mix that with this crystallization cocktail, which we'll talk about a little bit more, which consists of precipitants, additives, potentially detergents, and there are unlimited numbers of combinations that are used, and many different companies that sell different crystallization solutions. You mix those together, and then you carefully, very, very carefully flip over that watch glass and seal it on this Lindbro plate. So it's upside down, so we're able to have this vapor diffusion between your now half the concentration of the main well, which allows this droplet to slowly dehydrate because it has much lower concentrations of the main solutions, and it has a much, and so it is allowed to we twist the environment to allow for this slow dehydration. The other main method that's I think even more commonly used these days is the sitting drop method, which is much more you can automate much higher, which is why you see this is a 96 well plate with 2 droplets on each side, where we only have a much smaller number of hanging drop. So we have our classic hanging drop. This allows you to grow a lot larger crystals because you're using a much larger volume typically, because you need a nice big old drop that'll hang down. Whereas the sitting drop vapor diffusion method is very similar. So you have your droplet of your protein plus the crystallization solution on like a little table that's inside of your 96 well plate. And then you seal the whole plate with either, you can use kind of like an acetate sticky paper or the crystal clear packing tape that you might have used for moving boxes when you came to University of Michigan or when you moved with your parents. You can use that as well to seal these crystallization plates. So another method that you can use is this microbatch diffusion under oil. So this can be used for especially anaerobic proteins, so you can't have them have access to air. So you mix your protein solution with your crystallization solution, and then you actually put a droplet of oil over top, and then you can allow it to slowly diffuse into the gas of choice in your kind of anaerobic crystallization environment. There's also microdialysis, where you again have your protein plus crystallization solution, but you have a dialysis membrane to further limit the diffusion between the two. And this is one of my favorite, and I've never actually had the chance to do free interface diffusion, but the way this works is you actually have a capillary, like a capillary tube you may have used in general chemistry to like melt some samples to get melting points. Instead of putting, say, a powder to melt in, you instead pipette your crystallization solution in one end and your protein solution or your RNA solution into the other. And then you allow them to diffuse into each other, and you actually get crystals at the diffusion interface in specific points. So it gives you kind of a greater gradient of the concentration range between the crystallization solution and the crystal itself. And you can actually, depending on how these capillaries are made, just take that capillary directly to the synchrotron and diffract those crystals there. There's also been a lot of development on creating systems that are more amenable to like high throughput crystallization, such as there's a really cool microfluidic device that someone's created for crystallography. It's very interesting. So always looking for development in this set strategies, because if your crystals don't grow, then you have nothing to take to the synchrotron. So in all of these crystallization conditions, the idea is that we're trying to trick the phase diagram. So we have basically a concentration of our protein of interest on one angle, and then we have the precipitant or whatever additives that are in your crystallization cocktail on the other. And the idea is we want to move very slowly our protein solution into this growth / spontaneous nucleation point without getting into an aggregation point. So the idea is we want to end up in these supersaturated conditions that are able to create spontaneous nucleation, but not too much, because we don't want so many nucleation points that we get hundreds of crystals with different unit cells or different organization systems. We want a couple of nucleation points that are then allowed to grow and then slowly precipitate out of solution, rather than creating aggregates that then fall out of solution quite early and we're unable to effectively crystallize them. So this is one of the reasons why you try a variety of different precipitant cocktails, as well as changing the ratio of, say, your protein to your precipitant. So I was talking to you a little bit about precipitation cocktails. So there are 4 kind of categories of components in the crystallization cocktail. Specifically, we have inorganic salts such as ammonium sulfate, sodium chloride, KCl, magnesium sulfate is also very common. So these can encourage the formation of transient ionic interactions, as well as stabilizing some of the protein interactions. I've had a lot of luck with ammonium sulfate for our RNA binding proteins because it has a pretty high negative charge, so it can help stabilize those proteins that are looking for a negatively charged RNA. Another really common precipitant in a crystallization solution are these polymers, specifically polyethylene glycols and polyethylene glycol monoethers. These are actually, especially low molecular weight polyethylene glycol, are also a cryoprotectant. That means they help protect the crystals from radiation damage during the actual X-ray experiment at the synchrotron. There's also organic acids such as tartrate, citrate, acetate. These are also typically used in the buffer that you have in your crystallization solution, because pH is another very important factor in crystallization. So screening different pHs that may not be the ones you would expect for a protein in, say, a cell to exist. Specifically, like pH 5 can be more amenable to crystallization depending on the protein. But you can also use other organic molecules. So some groups have had luck with isopropanol or ethanol, but the volatility makes them more problematic for many applications. So it means that, let's say you do have a crystal, the first time you open that crystallization well, you have to harvest all of the crystals you have, because the concentration of those volatile substances is going to rapidly change the more you're interacting with that well. So one of the things that I think is really interesting to know about this is, for the most part, crystallization is a bit of a fishing expedition, because there's really no way for us to know the exact conditions in which a protein will crystallize, despite the fact that we've gotten crystal structures for a fairly large variety of proteins and a small portion of nucleic acids. There really is no way to say, you want to look at protein A, you should try these 5 conditions. So we have actually a variety of different screens that are used that you can kind of look at a wide variety of different conditions, or maybe a smaller variety depending on the protein that you're working with. So Thursday, I'll have a video. This is our crystallization robot. It's a Phoenix. It's very fun, and it allows this kind of tedious process to be automated in a really high throughput way, and it greatly decreases the amount of man hours that have to go into actually setting these trays. Because if you think about it, sitting down at your bench and pipetting microliter droplets into, so a microliter of every single crystallization solution and every single in your protein into every single well, that's 96 times 2, plus you have to fill all of those wells, and you're probably setting between 4 and 8 different trays because you want to try different crystallization screens. It takes a lot of time, and graduate students are some of the most expensive parts of running a lab because you have to pay us. We don't just get to work for free, which is nice for me. So this is one of the ways that we can really increase the amount of crystal trays we can set in a given time, and then all we have to do is go back and check them. So we have a 96 chamber dispenser. These are basically 96 syringes, and that syringe head goes down into these 2 milliliter clear cocktail stock boxes. These can either be purchased from companies, as well as you can make your own, depending on if you, say, have a protein that's been really, really happy with, say, ammonium acetate and citrate. You can make your own custom screens and then just keep them in these kind of blocks. So it'll pipette up all of those compounds and then put them into the 96 well crystallization plate, as well as it has a single channel nano dispenser, which is another very fancy syringe, which can put your protein or whatever molecule of interest into all of the single sitting drops. So this is one of the reasons why the sitting drop method is used a lot more than the hanging drop method, because it can be automated so easily. Whereas we can't do that with hanging drops, since each individual sample has to be pipetted onto a cover slip by itself. So I was talking about crystallization screening strategies. So these are the 4 most common types of crystallization screens. Typically, you'll see like random sample screens that go through this this matrix of additives, such as different ionic salts or some of those volatile liquids versus pH, versus the most common precipitant, which is polyethylene glycol. And as we vary those, we have this matrix of possible combinations. Random samples take a variety of these different concentrations within this 3-dimensional matrix and put that into your 96 well block. Whereas a footprint screen would say would move within a single, would hold one of the variables constant and then sample along the other axes. Whereas a grid screen are typically created after you've done a random sample. So you have a random sample screen, you've screened 96 different conditions, and in well, let's say E3, you get a crystal. So then you create a grid screen that varies those 3 different components that are in that positive hit to potentially further optimize that crystallization. And then you can also get the oversampled sparse screens, which are very similar to your random sample, but it increases the number of options within each of the random points of the matrix. So you'd say have 3 or 4 different concentrations that are similar to one another, but there's still overall, there's a matrix of different concentrations. So a lot of these are available commercially, and some of them have been developed and kind of optimized for specific types of proteins. But overall, as a field, it's kind of common thought that crystallography is still best viewed as looking at a random sample, because we really still don't know exactly what conditions work best for protein, or especially RNA and DNA crystallography. Yes?
Quick question. So, after you do a random screen, right?
Mhm.
Let's say you don't get any crystals, do we just quit at that point?
So typically, you would do two things. So you would try another random screen, because very few of these screens overlap. So typically you'd set like 4 trays in 4 different random screens. And if you don't get any hits in all 4 of those trays, you would simultaneously use whatever random screen that you have left. Usually you have 5 to 6 in a lab, and set a set of those. But then you would go back to that drawing board. You'd go back to your construct and think, okay, my protein has an intrinsically disordered domain, or it's predicted to. What if I remove that and make a new construct of my protein? So there are also a variety of different online servers that allow you to analyze the predicted structure, especially like with AlphaFold being available now. You can even use a more precise method. But if you look and see for like specifically like C-terminal and N-terminal domains on proteins are often disordered. So you can clip those off and create a truncated construct, or potentially you could try and crystallize a single domain of your protein instead of the entire complex, because especially larger proteins can be difficult to crystallize. If we're thinking about this from like a nucleic acid perspective, maybe your nucleic acid needs to be modified, so you can't do in vitro transcription, which is, I guess, the RNA equivalent of expressing in E. coli. It's a very quick and very far away from native method of creating a large amount of the macromolecule you're looking for. So you maybe would try and extract your RNA or put it into the environment where it could be further modified and therefore stabilized in the context. Also, one of the things people can do if their protein is not crystallizing is you can potentially add in usually it's antibody fragments, so Fabs, or other molecules that specifically bind to your molecule of interest, that you can potentially use their inherent crystallizability to force your molecule to also crystallize. So antibody fragments are often used for nucleic acids. Proteins that specifically bind blunt ends have been used as well, but there is a little bit of debate about whether or not that biases the structure you get. So it depends on the scientist you talk to about if that's a good idea or not. So in the video we showed you earlier, oh, it looks like my animations are messed up. Oh, so sad. In the video we showed you, like on the first day of class, there was a really cool robot that took pictures of your crystal trays every day, and they were like kept in this wonderful temperature controlled room. Very fancy labs have those. However, I would say the vast majority of crystallography labs just have a constant temperature room. This is a picture of all of the trays we have on one of the shelves in our constant temperature 20 degree C room. This is Lena's section. All of this is Meredith's. You can tell that she's been screening a lot of different proteins lately. And if I remove this image, here is the dissecting scope that we actually use to check our trays. So like in a biology lab, this is a dissecting scope, so it's not going to be the most powerful of microscope, but it does give you a lot of different objective views, and you can actually zoom in on your crystal with enough room to actually interact with the plate, because you will be harvesting your crystals while looking at it under a microscope, because you can't typically see your crystals because they're in these between 1 microliter to, I think the largest droplets I've seen people work with is 4 microliter droplets. So these are really, really small droplets that can dehydrate really fast. So here in this picture, I also have included what is called a crystallography magic wand, because crystallographers are very funny. And this is a magnetic tool that we actually use to pick up the pins on which we mount our crystals. So you would be manipulating this wand and your crystallography plate, and then you have this small storage door, which would be full of liquid nitrogen, and have your storage puck, which I will show you pictures of on Thursday, next to it. So it is a fairly delicate process, and it takes a lot of practice. But this is kind of the microscope setup that would most commonly be used. The other consideration is the fact that not all crystals grow at 20 degrees C. So 20 degrees C is great because it improves the evaporation rate, so it can potentially cause your crystals to form faster, but faster does not equal better, because by dehydrating faster, it often encourages aggregates and just your protein or RNA just falling out of solution and precipitating in a completely disordered fashion. So a lot of crystals are grown in the 4-degree room, so inside of a refrigerator, and a lot of those crystals don't like to be moved to 20 degrees. So that means sometimes sitting in a refrigerator and harvesting your crystals at 4 degrees, which can be a little bit harrowing, but luckily you don't have to do that all the time. So I've talked to you guys a lot about crystallography for a little bit. So we're about at halfway through the class. So I figured we could take a break for a second, and I have some beautiful pictures of the PRFA crystals I have crystallized in different crystal forms. And then when we get back in 5 minutes, we can start talking about actually how these unit cells and crystal lattices are formed. So if people need to take a drink, go to the bathroom, now would be a great time. Also, if you have questions, feel free to ask. Yeah?
So would changing your cocktail or the protein that you're using with the same cocktail change the physical layout of the phase diagram?
So it can. So it can push your, so changing the phase diagram, I mean, the phase diagram is the same regardless of your construct. It's just making it more amenable to being in that metastable state, that green zone. Because we want, so this is your supersaturated and creating aggregates. And then this is your just like in solution. So we want to be in this metastable state, that's supersaturated, but not quite precipitating out randomly. And so by varying the different concentrations and maybe trying different diffusion methods, so of your precipitation solution, and then this is your protein. And varying the concentration of these two will shift where you are in the diagram. So maybe instead of starting, at X concentration of your protein and Y concentration of your precipitant, you can move along the axes, but the properties of your protein and your precipitation solution will vary depending on your construct. So it can push it to be more stable or more likely to reside in this region. So more changing your temperature, which would change how quickly you're moving your dot along this this 2D matrix, is more likely to result in a shift. So if you see aggregates, that's a good sign in the sense that, well, I have something that at least is trying to sort of be ordered. So you could potentially move it from 20 degrees to say 4 degrees, or change your ratio to hopefully adjust your starting position and then the slow movement from, a low protein, low precipitant solution, so it would move into and stay in the metastable region longer, because that's when your crystal is actually being built. And then hopefully it will be moving into the fully crashed out solution in the context of a crystal, like we see here. This one's probably my favorite because it looks like a spaceship.
But changing the actual, what composes your...
So changing the contents of the precipitation cocktail is just something that's going to change the crystallizability of your protein. So we don't know when we start out what conditions are going to be the most amenable to creating an ordered lattice. Because a lot of times these salts or the crowding agents like the polyethylene glycol are really important for creating an environment where these highly ordered complex lattices happen. And without them, they're just so thermodynamically unfavorable, they never happen. Or they are happening too quickly, and so we get a bunch of different ones, which then form aggregates and then fall out of solution.
But that's unrelated to the supersaturation.
Yes, it's unrelated to the supersaturation.
So there's a quick question, right? So when we say a protein does not crystallize, right, does it mean that it doesn't have that metastable zone, or we just couldn't find it?
It usually means we couldn't find it, or it is not stable on its own. So specifically membrane proteins can be really difficult to crystallize because typically they need detergents or lipids to be stable. So one of the reasons that a protein cannot be crystallizable is it's too dynamic. So it's not able to be restrained in a lattice. So that's one of the reasons why we clip off disordered regions. But yeah, uncrystallizable typically means it's very dynamic. It may need a binding partner that is also not amenable to crystallization. I think it's better to say that it's not amenable to crystallization, or it has not, like it's uncrystallizable in our hands is much better, because there are so many different ways. There are infinite numbers of possibilities for these different crystallization conditions, and especially with the increase in novel methods for like membrane protein crystallization at specific scaffolds. We don't really know if anything is truly uncrystallizable. It's just within the scope of your experimental expertise. Yes?
If you like reconstitute a membrane protein into like a liposome or something, can you do crystallography with that? And like if you can, how does a liposome like not diffract? Or
So it would diffract. So that's the problem. And liposomes typically don't really like to crystallize. So typically they do nanodiscs, which are like a small like lipid raft that then your protein can be part of. Those are also used in NMR, but you would actually be getting diffraction because there's no way for you to silence that diffraction because they're still atoms. So they're still going to diffract. I think basically everybody is back. So I will keep on going. But this is where my notes are going to come in handy. So for the most part, things have been pretty easy so far when it comes to like crystallization. And everything gets a little bit more complicated when we get closer and closer to that atomic scale. So before I talk about different actual crystal contacts and the formation of these crystal unit cells, I do want to show you something. So all of these wells here are PRFA RNA thermosensor. But you can tell that there's different shapes of crystals as well as different sizes. So we have some microcrystals here, which I think look kind of like a starry sky, that underneath the polarizer, as well as these polarized RNA crystals. All of these are underneath the polarizer. Molecules with conjugated rings, so a lot of biomolecules, polarize light, which is how you're able to differentiate them from salts. That's why they look like pretty colors. They're actually just like clear crystals, but they're easier to see this way. But you can see there's a bunch of different shapes, especially we can see kind of this like four-membered kind of stack that's almost cubic in nature here. These are the same molecule, just in a different crystal form as my little spaceship-looking football or my little circular crystals. And you would think, because of the known crystal lattices, that they would always form in like cubes or hexagonal states. But the actual unit cell and the lattice that your crystal is forming doesn't necessarily have to match what we actually see in the wells. And quite interestingly, I thought that these might diffract better because they seem more ordered, because they're more cubic. These diffracted very poorly to very, very poor resolution, I think less than 6 angstroms, whereas my little football guys go all the way up to between 2.7 and 3 angstroms. So much better crystals, but aren't traditionally what you would think are better. Yes?
So are you able to preserve the crystals long enough to like look back at, or is it like a one-time?
So when you do your diffraction experiment, it is like a one-time because you've looped your crystal onto your crystallography loop. You've mailed it to Chicago or whatever synchrotron you're going to, and you're going to shoot it with X-rays. So for that individual crystal, it's a one-time thing. But you can harvest from the same well more than once. So maybe I scooped just this crystal and I left the other ones in there. But you do have to be careful because you've opened it to the environment, so you can kind of shift the evaporation state of that well. But you can save them. So if you're not sure, especially if you have some of these wells that are full of crystals, you harvest them all and you freeze them, and then you just keep them in your long-term storage store. And some of them you say use with a larger beam, and some of them you use with a very narrow beam. You use one that's like attenuated, so you have some time. Also, in a crystallization tray, they're stable for a very, very long time. So you have some crystals don't form for months, and some form within days. So it really depends on the construct. But once they're in a crystal, they're pretty stable, unless you start messing with them, and then they can fall apart. So we're going to talk a little bit about the properties of a crystal. So crystals are composed of these billions of identical molecules, but the key part of it is that they're in identically packed units against each other. So in this example, we have a 2D lattice, and each of these little parallelograms would be representative of a unit cell. So a unit cell is the repeating unit of macromolecules that you're able to translate in all 3 directions and get the same picture. So one of the things I think is really confusing about crystallography is a unit cell can contain more than one molecule. However, if you're crystallizing one protein, you have your asymmetric unit, which is that part of the unit cell that makes up the single repeating unit within all of the unit cells. So we have 2 of these birds, and each one of these would be considered an asymmetric unit, actually probably if we cut this in half. So we have the body of the bird and the head. Those 2 things together make up the asymmetric unit, so like here, because the pieces within that section make up the entire repeating image. So it's a little hard to grasp, but they can be related to higher order symmetry, so we'll talk about that later. This is twofold symmetry, so there's a point in this lattice where you can rotate by 180 degrees, and this molecule will be directly on top of the other. But the overall repeating unit is still this right there. Unit cell contents can vary from crystal to crystal, as well as the some crystals are able to form, or some proteins and nucleic acids are able to package in different ways, so they don't necessarily always have to look exactly the same. So we were talking about crystal packing. So here are 2 examples of crystal packing. So we have a tetragonal crystal, which has formed a cube here, but not necessarily. Not all tetragonal proteins form square crystals. And this is the specific crystal packing information. So this is the unit cell along with all of the descriptors you'd need to know about how this repeating unit appears in the crystal state. So right here we have the unit cell drawn in 3 dimensions, as well as the solvent content. So this is something that I found really shocking, is that actually crystals don't contain necessarily a majority of your protein or of your macromolecule. Crystals can have between 23 and 60% solvent content. So you can have a majority of solvent in your crystal, not necessarily your protein. So that can be one of the reasons why some proteins have much your diffraction patterns might be much fainter, and you would need more data to actually solve your structure because of the high solvent content. So these are two pretty moderate, this is a very moderate solvent content, and this is a very high solvent content. This is a hexagonal packing. We can see it has a different unit cell drawn here, and it actually has formed a hexagonal crystal, which is very beautiful. So when we talk about crystal lattices, so these are a formulation of your protein crystal lattice, which ultimately will be used to help describe your diffraction to other people, because there are a certain number of crystal lattices that chiral molecules and molecules in general can actually form. So only shapes with parallel edges can actually form a lattice. So you can't actually mix shapes, nor could you use shapes that don't have parallel edges. So like this pentagon here cannot form a crystal lattice, and neither can these like kite shapes. So only parallel edge things can create lattices, and it's a lot easier to think about lattices in 2 dimensions than it is in 3 dimensions. So they must be periodic. So all voids must be filled, and all tiles need to be oriented identically. So like here with these hexagons, they are oriented identically, and they are easily like tessellated and repeating. So the way we talk about crystal lattices in crystallography is through vectors. So each unit cell is made up of a B vector and an A vector. Typically, when we're drawing unit cells, A always goes down, B always goes out, and then eventually we're going to have C, which is going to go out of the page. So in 3 dimensions, it would be pointing at us. And then these are defined, they're also defined by an angle between your 2 lattice vectors. So we have 2 scalars, which is your A and B. In the case of a square unit cell, A is equal to B, and our gamma is equal to 90. So each vector has an angle between them, and this is how we were able to describe the way these repeating units occur in our unit cell. So we can see our repeating lattice here. So when we think about a unit cell, it's a combination of 2 things. We have our unit lattice, which defines the way they are repeating, along with our motif or our protein of interest. So this is from kind of the crystallography Bible. So there is a wonderful textbook by Rupp that this figure is from. But the idea is that you have your unit cell with your crystallized motif, and then it repeats within the lattice, and that gives you your plane group or your space group in 3 dimensions. So this lattice is periodic, and it has translational symmetry. There's 1 motif per unit cell, and so it is a primitive cell. So there's not more than 1 thing within your lattice. And you can see it's repeated, and the only symmetry we can apply to it is translational. All we can do is pick up molecule A and slide it over, and it'll align perfectly over molecule B. So we don't actually have lines in a crystal, so we just see these individual molecules in space. So we actually look for things called crystal contacts. So like you can see in between these protein molecules that there's these alpha helices that are creating a crystal contact and therefore stabilizing themselves. These are the things we're actually going to see. Or like this beta sheet interacting with this disordered region, or this unstructured region of the this alpha helix, so that these are interacting and stabilizing an interaction, and vice versa with these 2 unstructured regions. So this is what we actually see when we look at electron density from a crystallization, when we're looking at multiple electron densities in like our diffraction data. So we don't actually, so if we look at data from others and we don't actually see these contacts in 3 dimensions, it is a really good sign that that this isn't real. So the crystal contacts influence the structure especially on the side chains and flexible loops, and it can actually stabilize things that are not natural. So if we look at these flexible loops, you can't like infer a ton of things about them because they're involved in crystal contacts. So this will really affect your interpretation when we look at these crystal data sets. So earlier I talked to you about how in a unit cell there's more than just your asymmetric unit. You can have multiple things. And so this is what we call symmetry. So the full space group of your your molecule in crystallography is going to be defined not only by the lattice constraints, so like what kind of box is around it, and the vectors A, B, and C, and their representative angles. It's also going to be defined by these symmetry operators. So one of the most common one is this twofold rotation. So we have a twofold axis visualized here with this little diamond or football shape, which means that if we take molecule A and rotate it 180 degrees, it's going to perfectly overlay with molecule B. So this means that you will not change the motif, and they are consistent with the periodicity or the translational symmetry of the lattice. So this can't be true in one unit cell and not in the rest of them. This has to be just as periodic as everything else. So this is twofold rotation. So unit cells, unit cell transition along the B axis would generate a new twofold axis in the case of this unit cell we're looking at, because if we're moving this complex of two molecules over one unit cell, we also have a twofold axis in between the two molecules, because these are perfectly identical, that we have a second twofold axis. So it gets a little bit complicated when we think about the translational symmetry, and all of this has to be the same. So there are specific constraints for each specific lattice. So here we have a tetrad or a fourfold symmetry. So this is a 90-degree turn for your macromolecule. So we have molecule rotated 90 degrees, perfectly overlaps, 90 degrees, perfectly overlaps, 90 degrees again, perfectly overlaps. And again, this is going to create symmetry for your overall lattice group as well. These are all given different symbols. So that's why we had the little oval, and now we have the black square or diamond that indicates this. So this makes it a lot easier when you're looking at a crystallographic space group table for you to determine what are limiting your complex. So if we look at this tetragonal plane group, or P4, that has our fourfold symmetry unit, we can see in this instance that A and B are equal in length, and we have gamma is still equal to 90 degrees. You can kind of arbitrarily choose your origin in the context of unit cells, yes.
So when you say that there must be this kind of symmetry, do you mean in order for crystals to form, or in order for you to get diffractions from it?
So in order for this particular type of crystal to form, it must have these different, so when you're like classifying your crystal, so for it to be classified as a P4 plane group in 2 dimensions, these factors must be met.
So
Does that make sense?
Could a crystal form without any symmetry, like
No.
this? It couldn't?
Nope. So, well, it can form without any symmetry, but it has to have translational symmetry within from unit cell to unit cell. So the least ordered structure is P1, and that means you only have one asymmetric unit within your unit cell, and the only type of symmetry is translational.
Because I was going to ask, like, can you use computational models and say this protein could never have any of these kinds of symmetry, so the only kind of crystal you could get would be primitive?
No, because we don't know what kind of contacts it can have within protein-protein interactions, as well as interacting with the different salts or other molecules that exist in your crystallization complex. I think there are some bioinformatic analyses to tell you which ones are most likely. The important thing about this is it actually limits the amount of space groups that are possible for chiral molecules, and we're going to get to that soon. But this basically is really important for when we're actually looking at our data, because it can help us determine the space group, which will then let us move on to the next step of data analysis. So for a crystal to form, it has to have these regular interactions and form this ordered lattice. And although we aren't like part of that, we are witnessing it when we look at our data. And determining the space group is really important in the data processing process, the data processing pipeline, because having the space group is going to improve our ability to refine our structure and go through the phasing process, which we'll talk about on Thursday. Okay. So the important thing to see here is that these two partial, I guess like the unit cell would be visualized here. So we would see this small section, or this is a better image, of all of these fragments. And there's a fourfold axis where if we rotate the square around, it overlays. But in the context of the translational symmetry, there has to be this twofold axis as well. So just having one type of symmetry does not preclude you from having more than one type of symmetry. So the asymmetric unit, or ASU, as you'll see it in a lot of crystallography textbooks and the PDB, it is the smallest volume needed to generate the contents of an entire unit cell by applying crystallographic symmetry operators. So in this instance, if we're looking at half of a unit cell from the last picture, we only need this little section of the actual crystal unit to create the repeating unit within the overall lattice. So this is an important thing to realize, is it doesn't actually have to be the whole molecule in one piece, because we're looking at these in repeating units. So the ASU is this whole motif, but in our crystal lattice, because of where we have put the origin and where we've drawn the unit cell, this is the asymmetric unit that corresponds to our whole motif. So what is positive on the PDB is this is what's positive on the PDB, which is the ASU, but in one piece, rather than what you would see in crystal context, because that would look really silly. So if you know the unit cell and the plane and the space group, you can actually generate the unit cell contents. So if you want to, you can actually view a whole unit cell on the PDB using that structure viewer, and you can actually see how it crystallized with a 3D unit cell. And that can be really useful to see like what it might actually have looked like when you're analyzing that data. So there are 17 possible two-dimensional plane groups. So that's just the group within a 2D plane. Of these, only 5 are possible for chiral molecules because of the impossibility of glide planes, mirroring, and inversion centers because of chirality. The handedness of molecules really limits the amount of 2D groups that are possible. And if you are interested in really digging into plane groups a little bit more, I'm going to show you a really interesting Wikipedia page at the end of this lecture about plane groups or wallpaper groups, which can be really good at understanding like what actual like crystal symmetry looks like. So one of the types of symmetry that is disallowed, so not like rotational symmetry or translational symmetry, mirror symmetries or mirror planes are not allowed for chiral molecules. So just like you learned in OChem, the original copy of your molecule is not equal to a mirrored copy, because the mirror image of my left hand here is not the exact same as my left hand. Because we have these handedness or these chiral molecules, we can't have mirrors. So any achiral small molecule, so lots of small molecule crystallography, you will see mirror planes everywhere, but they are not possible. So if in your computations, your data keeps on saying like, well, it looks like we have a mirror plane, you don't. Mirror planes are not possible for chiral molecules. So in this instance, the real giveaway is these alpha helices. They're not in the correct orientation relative to each other. So you can see that this is not the exact same protein if you just perfectly mirror it. Here are the Bravo lattices that defined crystals. So a Bravo lattice is basically an infinite array of discrete points that are generated by discrete translation vectors and can be described in three-dimensional space by those vectors, where the vectors are also associated with an angle. So here are some of the these are all of the 5 possible 2D Bravo lattices that are available to proteins. So it's an infinite net of points generated by a set of translational operations that are defined by vectors. So in three-dimensional space, we have the addition of a C vector, but it's easiest to kind of look at, like for a rectangle, your A vector and your B vector will not be the same, but the angle will be 90. So whereas in like a rhombus, you have equal translation vectors and a different defined angle. Or the simplest one is a parallelogram, where you are not limited to the angle and your A and B are not equal to each other. So this would be what this could be considered so the parallelogram is the most simple of lattice. So that would be that primitive lattice, where you only have one asymmetric unit that is not described by any other type of symmetry. And that's all it is. So you can have a Bravo lattice, and then you have additional symmetry operators on top of those to define your full crystal contacts and your crystallography unit cell. So now when we move this into motif, everything gets a little bit more complicated because 3 dimensions adds a dimension. So we now have vectors A, B, and C, in which we have our motif in this 3-dimensional cell. And all of the same rules apply. The translational symmetry has to apply in all directions. So if we translate along the B axis or the C axis or the A axis, we still have to have that perfect overlay. So every individual unit cell is still perfectly repeating. So some important nomenclature for the unit cell, this is something you would find on the PDB for any depositive structure, is you would have the defined vectors A, B, and C, along with their corresponding angles. So we have gamma between A and B, we have alpha between B and C, and then we have beta between C and A. So these are the constraints that define our unit cell, and you can use your hand to help remember them, just like you would in vector calculus. So there are 14 3D Bravo lattices that exist that are possible for protein systems or any other chiral molecule. So these are all visualized here. So in 3D, there are 7 primitive crystal systems, and in combination with these 7 crystal systems, there are centering operators, which gives us these 14 unique lattices. And these lattices tell us how objects can pack into these arrays and can help us analyze our data. Because the more redundant our data is, the easier it is for us to analyze it. So these other symmetry operators, so if you have, say, the same molecule more than once in your unit cell, that increases the repetition and therefore the signal that you're going to get in your diffraction data. So so we have triclinic, monoclinic, tetragonal, hexagonal, rhombohedral, cubic, and then orthorhombic. And there are each subclasses beneath these because of, why is there only one transition? That's so weird. So in the primitive lattices, this prevents any centering and any other specific centering of the lattice. So these are all defined. So these do not contain more than one lattice point per unit cell. So you can't have repeating units within these unit cells. So you can have a primitive triclinic, tetragonal, cubic, which means that you don't have that secondary repeating element. However, if we look at centered lattices, we have the addition of a lattice point that improves or increases the symmetry. So these have face-centered, or so these are like fully centered, so you have a lattice point or another repeating or symmetry operator point in the center of the lattice, and then you can have face centering on any of the faces. So you have two planes, and then you would be centered along that plane, along with the face centering. And this can be possible in monoclinic, tetragonal, orthorhombic, cubic, and trigonal. So all of this feels very abstract, but as we talk more, it will make a little bit more sense. But I do want to talk about, once again, the 4 classes of symmetry operators that can be used to generate a unit cell when it's applied to an asymmetric unit. So all of these can be applied to generate a unit cell, but not all of them are allowed for chiral molecules. So as you can see, translation is possible for a chiral molecule, whereas, say, a mirror plane, which you can see between these two hands, is not possible. A dyad is only possible when it is in combination of what's called a screw axis, which I'll talk about, I think, on the next slide. But this, and an inversion, where it's inverted based off of a single point, is also not possible because of the chirality of our molecules. So one of the most, I think, powerful symmetry operators that we see the most commonly in specifically protein crystals and macromolecule crystals are these screw axes, which is a translation paired with a rotation along one axis. So this is I believe a 1/2. Yeah, so this is a twofold screw axis. So we're taking our protein of interest, our protein in our asymmetric unit, and we're translating it up half of a unit cell and over 90 degrees or 180 degrees. So these screw axes are defined by these different shapes as well. So we have our twofold, fourfold, and sixfold rotational axes, and then if you add these kind of motion indicating, as well as the threefold axis, that indicates it's a screw. So we also have that translation. So this would be half of a unit cell, 1/3 and 2/3 of a unit cell, half of a unit cell, a quarter of a unit cell, and then 2/3 of a unit cell. And that determines how far along the next unit cell you have to translate. So it's a translation and a rotation. And if this is confusing, that's okay. There are entire classes that are devoted to graduate students who do actually this analysis. This is confusing, but it is important to know that these are another way that symmetrically within a unit cell or within crystal contacts we can relate to different molecules. So there are 65 biological space groups. There are 230 possible space groups, but only a small subset are actually available to chiral molecules because of those limited amount of repetition that is possible. So this is great for us because it means that we have a lower amount of options that we have to screen through when we're actually analyzing our data. And just for interesting fact, P21, 21, 21, and P21 are the most common space groups that things are solved into, which is monoclinic, primitive, and then P21, 2 is orthorhombic, but still fairly primitive. So these have the least packing restrictions, which means that they are more likely to occur without those really specific packing interactions necessary. So the last thing I want to do for today, because we're right at time, is just quickly show you this, is that the space group frequency varies pretty widely, and they're like 90% of crystal structures have screw axes in these, 90% of the crystal structures occur in these particular space groups, and a lot of them have screw axes. But this, technically, every structure can be solved in P1, but that just makes it a lot harder. And by actually determining the exact crystal contacts, we can actually use our data more effectively and really narrow down what our asymmetric unit is, and therefore solving that electron density. The quiz will be posted shortly after this class. It'll be due tomorrow before class. It's going to be one question, so it shouldn't take very long. Please feel free to come to my office hours on Friday, and if that time doesn't work, the 2 times I have doesn't work for you guys, maybe I'll put a poll out there or something. I could do office hours by arrangement, if that would be better. So far, no one's come to office hours, and I've just answered all your emails during that. So if that would be better, please let me know. And I look forward to talking to you more about crystallography tomorrow. And by tomorrow, I mean Thursday. Yeah, no problem.
Do you want to do it after? Do you want to do it after the meeting?
So, there's a lot of different labs on campus that you can utilize it. I can just go ahead on different departments' list of faculty and do a keyword search. But honestly, if you watch someone, I don't know if you're busy at 10 o'clock on Fridays. I know I have class, so it's not always the best time for people. But going to the crystallography, the structural biology seminars can really tell you what labs do crystallography. So there's quite a few, like J.K. Kumar's lab does crystallography, the Kumas lab does, Jennifer Bridwell Grab's lab does crystallography. You'd be surprised how many there are. But if you're interested, just reach out to professors, and if they have space in their lab for an undergraduate, we're always looking for more people to help. So reaching out to a faculty member is always useful. Also, there is a crystallography unit, or lab activity in Biophysics 450. So if you want to get kind of access to just like what hands-on experience would be, you could potentially audit just that unit. In that class, because I'm guessing if you've already taken biochem and other classes like that, a lot of that class is review. If you've already done some higher division labs, but you could reach out to the professor teaching that class and see if you could audit that particular unit, because you actually set crystal trays and check them with lysozyme.
Yeah, that sounds really interesting. I think I'll do the seminar and see if I can maybe get into contact with that.
Yeah. Also, if you reach out to someone like Marcos or another chemistry faculty. Marcos Kumos. I meant to introduce myself at the beginning of this lecture, but I guess I'll do it at the next one. But that's the lab I'm in, along with Jennifer Bridwell Rabb's lab does a lot of crystallography, and Neil Marsh's lab does some as well. Who else do we collaborate with? Or Janet Smith. She's the crystallography wizard of University of Michigan. She's always a good person to reach out to. I don't know if she's taking any other grads this year, though.
So there's a potential I could be in the same library?
Potentially. It depends, like Marcos has room for more undergrads. We have 2 right now that are both juniors. So we typically look for students that are a little earlier on in their career, because it's just nice to have a student who sticks around to help with research.
Right.
But if there's another graduate student who's looking for an undergrad, there's always a possibility that he might end up doing some crystallography.
Yeah, that'd be great. Unfortunately, I'm also a junior, so.
Yeah, but that's okay.
But thank
but
you, yeah.
still, yeah, of course, reach out, shoot him an email, or any of the other professors that are.
Do you think you could